Hello students, in our today's session, we will be studying about discovery of DNA as the genetic material and a number of experiments that were conducted for the same, which includes Griffith's experiment, Avery, McCarthy and McLeod's experiment, Hershey and Chase experiment, which is covered in the chapter Molecular Basis of Inheritance. So let's get started. Students, let us see about the discovery of DNA. The modern understanding of DNA has evolved from the discovery of nucleic acids to the development of the double helix model. In 1869, Friedrich Miescher began working with white blood cells, which are the major component of pus from infections. He collected a lot of pus from the bandages at the local hospitals. He used a salt solution to wash the pus of the bandages. When he added a weak alkaline solution to the cells lysed and nuclei precipitated out of the solution. From the cell nuclei, he isolated a unique chemical substance to which he called nucleins. Chemically, these nucleins had high phosphorus content. Moreover, it showed acidic properties. Hence, it was named as nucleic acids. By the early 90s, we knew that Miescher's nucleins was a mixture of proteins and nucleic acids. There are two kinds of nucleic acid, the DNA, deoxyribonucleic acid, and the RNA, that is the ribonucleic acid. Few understood the significance since at that time inherited disease were thought to be caused by proteins. By the early 90s, geneticists knew that genes control the inheritance of traits, that genes are located on chromosome and that chemically chromosomes are mainly composed of DNA and protein. Initially, most geneticists thought that proteins are large, complex molecules and store information needed to govern the cell metabolism. Hence, it was at that time assumed that proteins cause the variations that are observed in species. On the other hand, DNA was thought to be small, simple molecule whose compositions varied little among species. Over the time, these ideas about DNA were shown to be wrong. In fact, DNA molecules are large and vary tremendously within and among species. Variations in the DNA molecules are different than the variations in shape, electrical charge and functions shown by proteins. So it is not surprising that most researchers initially favored proteins as the genetic material. But over a period of roughly 25 years, that is between 1928 to 1952, geneticists became convinced that DNA and not proteins was the genetic material. Let us study three important contributions that help cause this shift of opinion. Students, the first experiment that we will learn is about Griffith's experiment. In 1928, a British medical officer, Frederick Griffith, performed an experiment on bacterium Streptococcus pneumoniae that causes pneumonia in humans and other mammals. Griffith used two strains or two genetic varieties of Streptococcus to find the cure for pneumonia, which was a common cause of death at that time. The two strains that he used were the first one, which was supposed to be a virulent strain. It was smooth, it was pathogenic, and it was encapsulated and was called as the S type or the smooth type. Virulent, student, is that particular organism which is capable of giving you disease. And the other strain was a non-virulent, rough, non-pathogenic and non-capsulated, which was called as the R type. So the basic difference between the smooth and the R strain was that the smooth strain had a protective coating, extra coating on their cell wall, and the rough strain did not have that protective coating. Griffiths conducted 4 experiments on these bacteria. In his first experiment, I have just put the experiment in the graphical form in front of you. The first experiment, he took the rough strain and he injected it into an healthy mice. The mice survived. In the second experiment, he injected the smooth strain, that is the virulent strain, into the mice. The mice developed infection and the mice died. In his third experiment, he heat killed the smooth strain and he injected into the mice. In this particular step, he observed that the mice did not develop the infection and survived. Then he moved to his fourth experiment, where he used the rough strain and heat killed smooth strain and he injected into the mice. Now, as per his expectation, that is the rough strain and heat killed smooth strain, individually the mice survived. But when they were mixed together, unfortunately, the mice died. He was surprised at this particular observation. Griffith recovered large number of live S-strain bacteria from the blood of this last dead mice. In these four experiments, something had caused in the fourth experiment that he conducted, he realized that there was something that caused the harmless R strain to change into and changed into a deadly S strain bacterium. Griffith showed that the change was genetic. He suggested that the genetic material from the heat-killed S bacterium had somehow changed the living rough bacterium into the strain of the S bacterium. Griffiths concluded that the R strain bacterium must have taken up something to what he called as the transforming principle from the heat-killed S bacterium, which allowed the S strain to get transformed into the smooth-coated bacterium and become virulent. Students, in 1944, after some 10 years of research and experimentation, US microbiologist Oswald T. Irby, Colin M. McLeod, and Macklin McCarty, all at the Rockefeller University in New York, first evidenced the proof that DNA is the genetic material, or the transforming principle that Griffith spoke about in his experiment. Students, how they performed the experiment is they purified the DNA, RNA, proteins, and other materials from the cell-free extract of the S strain and mixed them with the heat-killed S strain and R cells separately, living R cells separately, to confirm which one of them could transform the living R cells into S cells. That is, they individually took a protein, RNA, DNA, the sugar coats that are responsible, they mixed it with the S strain and the living R strain. And you would see that if only sugars were added, there was no transforming principle. If only RNA was added, there was no transformation happening. If only proteins were added, no transformations occurring. Only when DNA was added, there was transformation. This they further confirmed, and the mice died. This they further confirmed by discovering that protein digesting enzymes, proteases, and RNA digesting enzymes, RNases, did not affect transformation. So the transforming substance was neither the protein nor the RNA. When they added the DNA digesting enzyme, there was inhibition of transformation. The experiment proved that the transforming principle is DNA, but all biologists were not convinced. Students, have a look at this image. It will provide you a better understanding of the experiment. That is, there are two sets. For example, in this case, they took the rough, that is the non-virulent S type, and the heat-killed smooth virulent S type. So they took the non-virulent R type and the smooth virulent S type together, and they mixed it with a protein digesting enzyme. So the proteins were digested, and when that was injected into the mouse died. But when they mixed the same thing with DNAs, that is DNA digesting enzymes, in that case, when it was injected into the mouse survived. which confirmed that DNA is required for transforming the smooth, the virulent, non-virulent R type into the smooth type. Finally, Alfred Hershey and Martha Chase in 1952 proved that DNA is the genetic material and not proteins by using bacteriophages. Students, bacteriophages are viruses which are capable of infecting bacteria. Hershey and Chase worked with bacteriophages. Now, bacteriophages are composed of two important components. One, the DNA, and second, the proteins. Now, they used radioactive phosphorus P32 in a medium for some viruses and radioactive sulfur S35 for some other viruses. They grew some viruses on a medium that contained radioactive phosphorus and some on the medium that contained radioactive sulfur. Now you need to know here that sulfur is present in proteins and not in DNA, and phosphorus is present only in the DNA and not in proteins. Hence, the viruses that were grown in the presence of radioactive phosphorus contained a radioactive DNA but not a radioactive protein, and because DNA has only phosphorus and it doesn't has any proteins. And similarly, viruses grown on radioactive sulfur contained radioactive proteins but not radioactive DNA because DNA does not contain sulfur. Now, these radioactive phages were allowed to infect normal E. coli bacteria which were grown on medium containing normal phosphorus and sulfur. Now, as per the bacteriophage's life cycle, it normally injects its genetic material into the bacterial cell. So now we have to identify what the genetic material is, whether it's the protein coat or whether it is the DNA. So the viruses were allowed to infect the bacteriophages, that is the bacteriophages proceeded with the infection. The viral coats were removed with the help of centrifugations. The bacteria which were infected by viruses with radioactive DNA were the ones indicating the DNA was the material that was being passed on from the virus to the bacterium, because the radioactivity was discovered inside the cell. That is, when the supernatants were detected in the case of the cells that were infected with the radio-labeled proteins, where the sulfur was detected in the supernatant and not in the bacterial cell, and the ones infected with the radio-labeled DNA, the radioactivity was discovered inside the cell and no phosphorus was detected in the supernatant. This indicates that the proteins from the viruses did not enter into the bacteria. Therefore, the DNA is the genetic material that is passed from the virus to the bacteria. In other words, sometimes after infection, radioactivity for phosphorus and sulfur was tested. Only radioactive phosphorus was found inside the bacterial cell, indicating that the DNA is the genetic material and the bacteriophages infect its genetic material into the bacterial cell. Thus, this Hershey's and Chase experiment gave the final evidence to prove that the DNA is the genetic material or the transforming principle that Griffith spoke about in his first experiment, and thus led to the discovery of DNA as the genetic material. Students, this image is called as the DNA timeline. You can have a look at here about the scientists and the number of experiments that were conducted to finally conclude that DNA is the genetic material. You can pause the video here for a while and have a look at the chart. Thank you students for participating in this lecture. We will meet soon again with the next topic. Till then, take care, be safe and keep learning.